strategies to transform animal cells. So basically, 4 types of methods have been used to transform animal cells. And broadly, they can be categorized into biological methods or non-biological methods. Non-biological methods included like physical transformation, chemical mediated transformation, and biological mediated transformation can be discussed under the headings like transduction or bacteriophage. Now, if we focus on some biological agents that can be used for animal cell transformation, so first we can consider on the process of transduction. In case of transduction, it is viral mediated transformation that we can take the viral particles as an infectious agent or as a vector, and then we can transfer our exogenous DNA or the target DNA into the phage particle, and then it can be used to transfer DNA into the target cell. So this is one of the biological method that can be called it as the transduction. In case of bacteriophage, we can insert our exogenous DNA into plasmid that can be used to transform bacterial cells. And then bacteria can be used to transform animal cells. And this process where bacteria can be used to transform animal cells, it can be simply called as bacteriophage. So these are some biological methods that are phage mediated or bacterial mediated that can be used. Non-biological method, first one, you can say that chemical mediated transformation. In this case, DNA, a foreign DNA, it can be complexed with some positively charged compounds like lipophilic vesicles. They can be used that are positively charged. So our DNA, it is encapsulated or contained within lipophilic particles or vesicles. तो ये जो वेसिकल हैं ये पॉजिटिवली चार्ज होते हैं और सेल्स की जो सरफेस है ये प्रीडोमिनेंटली नेगेटिवली चार्ज होती है. तो पॉजिटिव DNA complex will interact with negatively charged cell surfaces. So interaction is there, and by the process of endocytosis, the foreign DNA, it can be inserted into the cell, and this technique, it is called as chemical mediated transformation. Then another method, fourth one, it is the physical transformation that naked DNA with the help of certain chemicals, even the use of particle bombardment technique, electroporation, it can be transferred directly into the animal cell. So it can be explained with the help of diagram. These are the 4 techniques or 4 principal mechanisms by which DNA can be introduced into animal cells. As I told you that, like we can start this physical transformation that by using electroporation, particle bombardment, foreign DNA, it can be directly inserted into the nucleus of the animal cells. Alternatively, this foreign or exogenous DNA, it can be complexed with positively charged compounds like the lipophilic vesicles. And after encapsulation, these positively charged DNA complex, it will interact with negatively charged cell surfaces. And by the process of endocytosis, this complex, it is being engulfed and then transported to host nuclear DNA where it can integrate. So these 2 techniques, we can say that they are non-biological one. Then biological techniques, First one, phage mediated, that foreign DNA, it can be encapsulated within phage particles, and then it can be used for the insertion into the host cell. And then another bacteriophage technique is that bacteria can be used that contain a recombinant plasmid, so bacteria can infect the host cell. And then it also results in the integration into the nuclear genome. So these are 4 principal mechanisms by which DNA can be transferred or introduced into the host nuclear genome.